So thanks so much for coming to my session, and thanks for giving us attention after a long week of a lot of really interesting talks. So I'm CEO of Abterra Biosciences. We're a company that we spun out of UC San Diego from the computer science department about 10 years ago. And so like many of you, machine learning is kind of an important part of our DNA and an important tool in our toolkit. But full disclosure, I'm not going to go in detail into our machine learning methods in this talk. But I hope you still find it pretty interesting. So our company does antibody sequencing and discovery services. So we like to say if you have an antibody that you like, whether it's a single B cell or a phage library or a hybridoma or bulk tissue, we use NGS to sequence it. If you have antibody protein, we can sequence that as well. We've been doing it for thousands of antibodies, and so we have a lot of experience in those hard cases. So if you need an antibody sequenced, there's a good chance that we've already seen something like it and we can help you out. If you don't have an antibody that you like, then we have the ability to help you find an antibody that you like. We've developed a platform called Alicanto, and that's what I'm going to talk about using in this case to mine convalescent patients for neutralizing antibodies to SARS-CoV2. And I'm also going to take the liberty of assuming that you've heard of that virus and you know that we're in a pandemic, and I can kind of skip over the basics. So we've known for 100 years, what serum antibodies do in response to challenges like infections and vaccines. We know that after kind of an initial exposure, there's a bit of a delay, but then we see antibody titer increase in serum, starts to wane a little bit, but then upon boost, we get an increased response that persists for quite a while. But until recently, we didn't understand what was happening under this hood, kind of on a molecular level. What were the clones doing that were contributing to the serum response? So it's only really been in about the last 10 years with immunosequencing that we've really seen what's happening under the hood and understood what different clones are doing in response to the challenge. So we've learned that this immune response is vast and fairly complex. So we see B cells changing across time, changing across different boosts. But we've kind of missed that first question of the serum antibody titer that we were observing 100 years ago when we were looking at people that were being exposed to viruses. So that's why we built our Alicanto platform. It allows us to basically connect the dots between what we see being expressed in the B cells and what we see being present and measured in serum. And when we started to look at Alicanto, and we've done this on over 100 projects at this point, we've realized that actually a lot of those B cell sequences that we see in the repertoire are not being significant contributors to the serum response. So Alicanto is our platform for mining the serum antibody response, harnessing that last step of in vivo optimization to identify good antibodies for your projects. So serum is actually, kind of a really great source of antibodies. So first of all, serum is where antibodies hang out. They are abundant and accessible in that compartment. There's 11 orders of magnitude more antibodies in blood than there are B cells. Now I know they are smaller, but 11 orders of magnitude is massive. That's the difference between grains of sand in a bucket and grains of sand in all the beaches on Earth. So there's a lot of those suckers. And that means that on kind of a very small volume of blood, we can potentially screen millions of different antibodies that are at a detectable level. So that's what Alicanto does. It gives us access to those serum antibodies. But also, we have an approach that allows us to sequence everything. So we look at every antibody that we encounter, not just the hits at the end. And that allows us to make sure that we're sampling the diversity of the immune response, which is important because our immune response is not uniform. We have some very large dominant clones and some very rare clones. We want to make sure we capture all of those. And it also allows us to do hit expansion, so we can find relatives of our screened hits that have other properties that we like. So kind of at its simplest form, Alicanto starts with a volume of blood, which we fractionate into IgGs present in serum and all of the different populations of B cells that are present in PBMCs. But where that blood comes from, it doesn't actually matter. So we built Alicanto to be very flexible. Anything that has a backbone and a jaw has an adaptive immune response, and we can pull antibodies out of it. We work with a lot of species, but most commonly we work with rabbit, single domain antibodies from camelids like llamas and alpacas, and then also antibodies that are present in humans. Now, the antibody proteins have to be analyzed using different technology than B cells, of course. So we use mass spectrometry to analyze those antibody proteins directly. And then we use next generation sequencing to analyze the B cell repertoire from that volume of blood. Now, in the hyperimmunized animal case, Alicanto is useful for capturing what antibodies are giving you the serum titer that you're seeing. Whenever we're immunizing an animal, we're monitoring that serum titer to say whether or not the immune response is successful. So kind of the key difference of Alicanto is that we don't throw away that serum and then go looking for a B cell that makes an antibody that gives us the reactivity we want. We actually look directly at that serum. But in humans, that serum antibodies have kind of a special purpose as well, and especially in infectious diseases. We know that the humoral immune response, and antibodies in particular, are critical for how we overcome viral infections and protect us against the next one. So we used Alicanto in the virus that's been plaguing our lives for the past 3 years. And we looked at 3 patients. And at this point in the pandemic, it's hard to find people that have a tidy immunization and infection history. But these are the 3 that we got. The kind of key things to pull out from these exposure histories is that all 3 of our patients were both immunized and both immunized and naturally infected, but in different orders and with different variants. So AB2 was infected and also immunized with wild type version of the virus, so they were not exposed to any of the newer Omicron variants, whereas AB5 and AB6 were immunized with wild type, but they were infected with Omicron variants of the virus before blood was taken. The other thing to note is that we know that AB6 didn't actually have seroconversion after their immune response, so there was no IgG to the virus after their boost. So the infection with Omicron was sort of like a prime, not necessarily like a boost of a previous response. And all of these were not, blood was taken 3 to 4 months after their kind of last exposure to the virus, so it was quite a bit after their infection or vaccination. So from those individual patients, we collected blood, we fractionated into 2 different compartments that we're analyzing. So from blood, we took total IgG, and then we pulled out 3 different fractions that have reactivity to different parts of the spike virus. So we were mostly interested in antibodies that bind to receptor-binding domain, or RBD. That's how the virus gets into our cells and infects us. So we first stuck RBD on a column, pulled all the antibodies out that bind to RBD. We wanted to make sure we had everything, so all of the flow through that column, we purified on a second column. We call that RBD times 2. So that's everything that didn't catch the first time, we got it the second time. And then everything that didn't bind to RBD must be binding to some other part of spike. And so kind of our third step in this workflow was to pull out any spike binding antibodies that didn't bind to RBD, but bind to other places. Or to different structural epitopes, because spike is a trimer, then the RBD was a monomer. And in this case, we purified only on Omicron B11529 variant. So again, this was a variant that 2 of the patients had maybe seen, but 1 of the patients had not seen. On the B cell side, we did something very simple. So we just took total B cells. We didn't isolate any particular population. And we just did antigen enrichment. So any B cell that reacted with spike protein of B11529, we pulled out for analysis with Alicanto. On the serum side, we applied our standard mass spec process. I'm not going to go into detail on this, but we have publication about how we do this. It's kind of a standard bottom-up approach. We digest those proteins into individual peptides. We analyze them by the mass spec, and then what comes out is a mass spectrum that's associated with each peptide that we got from our polyclonal. And on the sequencing side, we did single cell sequencing. So what comes out of that is a big database of tens of thousands of sequences, but we have paired heavy light chain information on the B cell side. And that forms the input to our Alicanto platform, is the mass spectra and the NGS sequencing data. So under the hood, Alicanto basically maps the mass spec data onto all of those antibodies in the B cell repertoire and tells us which ones are being secreted in serum. And this is kind of what it looks like under the hood. So whenever we run an Alicanto report, we generate one of these, when we run an Alicanto project, we generate one of these reports, which has all of the evidence for how we pick these individual candidates. So what I'm showing is kind of a genome browser view of an individual antibody that was detected in this project. So along the top, we have the amino acid sequence annotated with CDR1, CDR2, CDR3, all of the mutations that have been accumulated in this antibody with respect to germline. And then all of these individual blocks are peptides that are mapping to this individual antibody. So they were observations that we found in the polyclonal that we pulled out from serum that match this individual antibody. And you can see that they're different colors because we use different enzymes. So we get lots of overlapping peptides. We don't have kind of big gaps in our sequence. And they cover the whole antibody. So they cover areas that are very conserved and shared across antibodies. But they also match to areas that have mutations and are fairly unique to this individual clone. So this is an individual antibody, but most of the time in this talk, I'm going to be talking about clones. And as a smart man at a previous conference said, we really got to talk about what we mean when we say clone or clonotype, because we all mean something different. So when I say clone, I'm talking about the antibodies that all share the same amino acid CDR3 sequence. It might not be your definition of clone, but just, the disclosure slide. And then we say that a clone is present if we get good proteomic information, proteomic evidence covering and supporting that particular CDR3. So in this project, we did single cell sequencing. We had our 3 different patients. So we sequenced all of those spike-specific B cells that we pulled out, and we found between 30 and 45,000 different paired antibody sequences from the B cells. When we collapse those into clones, we don't actually get a lot of collapse. The repertoire is quite sparse. We don't have a lot of antibodies that share the same CDR3, so it is quite a bit of diversity that we captured there. And then when we pull out the ones that are very high confidence present in serum, we find that there is quite a down selection, and there is quite a lot of difference across the different patients. So by far the most kind of productive and diverse patient was AB5, and we found about 90 different clones present in their serum across the different fractions. AB6, who is the individual who had, the latest infection was kind of like their prime immunization or prime exposure, had less clones detected against this variant. And then AB2 only had 2 clones pulled out with high confidence from the RBD purification fraction. But I guess I just want to highlight again that AB2 was not infected with Omicron. So anything that we pulled out that was reactive to the RBD happened to be cross-reactive with this newer variant that this individual had not been exposed to. So kind of our first question was, this is a pretty big down selection, right? There's a lot of sequences in the background, and not all of them are detected at a high level in serum. And so are there differences about the serum antibodies compared to that background repertoire? So the first thing we looked at was just, are they more mutated? And they are. So in the background repertoire, because we didn't do any isotype sorting, we didn't do any specific population sorting, we were only looking at antigen binding. 70% of those sequences had no mutations and completely matched the germline. Whereas when we looked at the serum identified clones, they tended to have, only 10% of those were unmutated, and they largely had a nice profile of mutations. This is, because of how we set up the experiment. On the serum side, we pulled out only IgGs from serum. And so we have a bias on that side for a particular isotype. And in fact, that's what we see when we look at the isotype distribution of the background repertoire versus the part of the repertoire that we see in serum. So on the left side is the background repertoire. It shows that 80% of our sequences were IgM isotype and a teeny fraction were IgG. Whereas if we look at our serum detected clones and look at their isotype in the repertoire, that ratio flips. So 70% of the antibodies that we detect in serum are found as IgGs in the repertoire, but there are 20% that are only observed as IgMs that then class switch and are secreted as IgGs in serum. So it's kind of interesting that there were some IgMs in the B cell repertoire that then class switched and we detected and had kind of good mutation loads and were detected in serum. So we picked kind of a smattering of about 24 different antibodies to test across the patients, across the different purification fractions. And we wanted to do basically low-res epitope mapping. So, how many of the guys that we pulled out against RBD actually bind RBD versus other places on spike. And our proxy for other places on spike was the N-terminal domain, so just NTD. So for every antibody, we tested it against spike, RBD, and NTD. And then I segmented it across the different individuals that we found. And so kind of not surprisingly, RBD binders, because that's what we were really looking for, were common to all patients. And they had a range of affinities from kind of single-digit nanomolar all the way up to 170 nanomolar, so kind of a range of affinities. 3 of the non-RBD binders we could localize to NTD. And then 2 of the binders we couldn't actually localize to any particular domain, so they bound to spike trimer, but then they didn't come out against RBD or against NTD. And so we're following up with kind of higher resolution epitope mapping to see where these guys bind. And then interestingly, 3 of our RBD binders, again, only appeared as IgM in the repertoire, and then class switched and became IgGs in serum. So I mentioned that our repertoire was fairly sparse. So if you wanted to try to do hit expansion and find other candidates that had the same CDR3, there was limited ability to do that. So we also did bulk repertoire sequencing on basically everything else that we found in these individuals. And so we were able to fill out some of the trees of some of our candidates a little bit better. So what I'm showing here is kind of a standard lineage tree. Each of the blocks is an observed antibody sequence from the B cell receptor repertoire. The green guy is one of our candidates that we found in serum. And so by doing bulk sequencing, we could fill out this tree and find 7 other candidates that are in the same clone as our tested candidate. In the tree, the germline is the germline rearrangement because these guys all share the same CDR3. And then all of these intermediate nodes are the intermediate kind of inferred sequences on the way from the germline to the observed sequence. So in this case, our candidate had 30 nanomolar binding, and then by doing bulk sequencing, we could find other candidates that had better affinity. Again, as I said before, though, there is still limited utility in doing this. So because the immune response is this exponential distribution, there are tons of guys that are very rare and don't have any family members, even in the bulk sequencing. So even after bulk sequencing, about 1% of clones had another family member, even just one family member. So most guys don't have another sequence that has the exact same CDR3. So when we were trying to do hit expansion, we had to zoom out a bit and not just look at exact CDR3 within a clone, but look at clone clusters where they have sequence similarity. And the hope is that you have this kind of small but very common lineage tree from a given CDR3 where you don't have a lot of observations, but maybe by doing sequence similarity or by looking at physicochemical properties of that CDR3, you could find another tree that has a lot more fruit to pick. So we took that same candidate, we did that zoomed out clustering just on sequence level. In this case, we took all CDR3s, we clustered on 80% identity in the same germline V gene to get clone clusters. And we found kind of a cluster of 8 other CDR3s, 7 other CDR3s that we could then mine for other candidates that had similar specificity, but hopefully other properties from our candidate. So our data set was definitely kind of a small one compared to the compendium of knowledge that we now have against coronavirus. And And so instead of just looking at our data, we thought, why not just look at all the sequences that other people have found as well? And so there's a great resource called COVABDAB, which has coronavirus binding antibody sequences. And so we pulled out all of the sequences that people have reported that bind to SARS-CoV2 that are from human, and they have binding information about, where the epitopes are that they bind to. And we clustered them using the CDR3 sequence similarity clustering with our candidates. And about 300 of our B-cell receptor sequences did cluster with public sequences. And then 3 of our tested 24 candidates also cluster with public sequences, which was kind of surprising because we don't often see our sequences in other places. But in this particular hairball, so this is kind of one cluster of sequences that had both sequences from this project, but also sequences from the public database, was kind of interesting because I'm highlighting the 2 antibody candidates from our project, which are the mauve triangles. And then the green and the mustard are the public clones that had within 80% identity of our particular hits, or within the same click. And these ones are annotated as having binding to the same targets as our MABs 10 and 13. 10 and 13 bound to RBD, and the green clones were reported as also binding to RBD, whereas the mustard clones only resolved to spike. And then it also clustered with other sequences in our repertoire, as I showed in the previous slide. But this becomes kind of a fuller picture of those individuals, and it was exciting to see kind of the same convergence that other groups have observed across patients in this virus. So kind of our second question was, to what extent do we see binding across the different variants? And this was just in ELISA. And so we took those same 24 candidates and we tested them against the wild type, the B11529 Omicron variant, and then a newer Omicron variant, BQ11, which none of the patients had seen, to see whether or not there was cross-reactivity across kind of the variants they had been seen, and then also the newer variant. And so kind of not surprisingly, all the antibodies bound to wild type. 2 of the 3 NTD binders also were pan-variant. I think this is consistent with other groups have seen. So because NTD is not necessarily kind of a site of neutralization, it's not under as much pressure, and so it's more conserved. And then 3 of our 8 RBD binders were shown to be pan-variant, and an additional 4 were cross-reactive between at least one of the Omicron variants. So that was pretty exciting that these kind of could recognize epitopes that were conserved even on newer viruses. And then kind of the last step was just to see, well, okay, they bind to the spike protein, but do they actually block ACE2 binding? And so several of our RBD binders, we just in vitro measured blocking of ACE2 binding, and we found that 6 of them did show ACE2 blocking, so that was pretty cool. And these were derived across all of the different patients. So it wasn't just AB5 who had kind of the most diverse and strongest immune response, but also AB2, who had never even seen an Omicron variant, had good neutralizing antibodies that just happened to kind of react with the Omicron variants. So that's it. So I just want to summarize that we developed Alicanto to be a platform to discover diverse antibodies that are present in serum. And so if you have either a target or a patient sample that has an interesting serum phenotype, we'd love to talk with you about how we could help you find the antibodies that are present in that. And I also want to just thank our team at Abterra Biosciences. We do a lot of work in human, but we also do a lot of work in alpaca and llama, and it's always cuter to have them on the slide. If you have any questions, I'm happy to take them, or you can visit us for a very short time at booth 525 down in the exhibit hall. Thanks so much for your attention.
Great. Thank you, Natalie. I've been waiting for a platform like this that would map IGs in the serum to the B-cell receptors, so that's fantastic. Oh, so you cannot hear me?
I got like the second half of that. Was it a question?
I thought the platform is something that I've been waiting for. Any questions from the audience?
Yes, hi. Fantastic talk. Thank you so much. I saw that you are actually looking at individual serum. You took one individual and AB2, 5 to 6. I was wondering whether you can do it for a group of patients, for example, and try to see a pattern in a certain group of patients compare, let's say, for healthy control. Thank you.
We haven't done that. So the process, so part of the limitation of comparing to healthy controls is that we need to be able to pull out antibodies from serum against the target. So if it's someone that hasn't been exposed to SARS-CoV2, then the chance to kind of pull out anything from serum is pretty limited. So is that what you mean?
I'm not talking about SARS. I'm talking in general, like a group of patients that have some type of tumor or some type of autoimmune disease, and we want to see the type of bias repertoire which is present in the serum compared to healthy. I mean, just to have an idea what happened in that group of patients. Is that possible?
I think it might be. So I know that people are doing that on kind of the B-cell receptor repertoire basis. Because the serum antibodies are kind of such a small subset of that, it's not immediately clear to me how we would do that comparison across, cases and controls. I do think the opportunity to mine the serum of healthy patients for tumor antigens is very interesting because, those people often don't even present with disease and they have anti-tumor antibodies. So those people are very interesting from a discovery standpoint, but we haven't looked at kind of population comparison.
Is it possible?
Anything's possible, maybe.
Okay, sure, please. One more question.
Hi, yeah. So you're taking a limited number of B cells from a patient to do your NGS analysis. Have you tried doing N of 2 or N of 3 to see how that changes from an individual patient, to see whether the serum antibody and the NGS data sort of marry each time?
And being like multiple samplings from the same patient for the B cells. So we haven't done that. In general, what we get from humans is just one time point. So we haven't done a lot of like kind of longitudinal sampling from individuals, but we do that a lot in animals. And I'll tell you, it's very species specific, kind of how the B cells change. So in rabbits, for example, a lot of times we'll see the same clone appear a lot of times. So the guy that's present at the end of a 3-month immunization often arose at the very beginning and just kind of matured through the whole process. Whereas in llamas and alpacas, we don't see that. Things go up and down, and was that your question, or did I go off?
Yeah, I'm just guessing whether the NGS data set from a sample is repeated when you take another sample from the same patient.
Oh, I see. Yeah, so we have looked at kind of replicates, but only from kind of the same bleed. So we haven't done kind of we haven't done the longitudinal, if that's what you're taking other samples from. But in terms of kind of overlap across individuals or from the same sample, if we just aliquoted it and analyzed it 2 different times, we get pretty high overlap. So depending on how late in the process you start, it could be as high as 80% overlap. If you're taking kind of like, splitting the cells and doing 2 different extractions and everything, then it can be a little bit lower.
I think, Natalie, Mike, I think you were asking whether it's really coming from the same bleed, right? And you just wish to have, let's say, 2 readouts from the same sample. So you have the basic replication, so you can see whether there's some technical, factors that gets introduced in, the quality or the reproducibility, basically, of the data that would then move forward for the further analysis. From the same bleed.
So technical variability that could influence the downstream results. Yeah, I think that is a case. So like I said, if you take kind of like, even if you just like sequence the same library 2 times, you don't get 100% overlap across those individuals. And I think from a biological standpoint, the antibodies that are present in serum are not necessarily the ones that are present in blood. So maybe a more ideal sample would be bone marrow, for example. We don't always have access to that. So I think the hope is that we're catching something similar to what's secreting the antibody in bone marrow.
Okay. So one last question before the break, because you mentioned that you were doing single cell sequencing for the B-cell receptors. Are you also getting the transcriptomic readout for each cell?
We don't do RNA-seq, we just do the antibodies.
Okay,
sequences.
because I think it would be interesting also to do that because you have that capability to map the ones that have already been secreted, whether using omic data you can identify the B cell maturation stage and identify those populations that really correlate to those that secrete. And I can also imagine using surface ID on some of us and also using the diffusion models of Regina to have an interesting screening like that. Well, thanks a lot, Natalie.
Thanks.